So knowing about crossing over, it turns out that the chance that crossing over happens is something that we can predict with pretty good accuracy. And it sort of also relates to the fact of how we define the probability of these things happening. So there's a unit called a genetic distance, or some of these units here, map unit, centimorgan, and these are units of distance that aren't physical distance so much. They're more of like a probability distance. As we'll see, they relate a little bit to physical distance, but they're not one-to-one with physical distance. So early on, this would be the way of mapping the locations of genes on a chromosome. So if you found a new gene, these are fruit fly genes. If you found a gene, vermilion eyes, and you discovered a fly that's got these bright red vermilion eyes, and you wanted to sort of find out what chromosome that's on, you would start crossing in your vermilion-eyed mutant with known mutants at other genes. So you would see what happens if I cross vermilion with yellow. So yellow body, vermilion eyes, I can make a cross of those, and you would notice at some point that the chance of the recombinant offspring in there was a little bit lower than you'd expect for unlinked genes. You'd start building the case that they were both located on the X chromosome. And by doing crosses after crosses after crosses, you'd collect a lot of data, and you could determine that about 30% of the time, crossing over happened between yellow body gene and vermilion eyes gene. So crossing over happened not completely randomly between these two, but predictably as far as their physical distance between them. So 30%, that's fairly likely for crossing over. But if you had vermilion-eyed fruit flies and miniature wing fruit flies, you would see that the crossing over between them is only about 3%. So much less likely. 3 out of 100 offspring would show evidence of this crossing over. All right, so like I said, map unit is, I think I prefer to use this term, map unit. There's a unit that it's got this kind of SI unit feel, centimorgans, which is just, it's all the same kind of terminology. So centi referring to the fact that this is a percentage, so sort of 100% would be, I guess, 1 Morgan. Good Morgan. All right, so crossing over, more likely over greater distance. Let's see if we can illustrate that a little bit. So if you've got long chromosome arm, I'm going to just simplify this down and keep us with a couple of chromosomes arms here. So we have the yellow, let's say we're looking at fruit flies here. So you could have the yellow with the mutant Y allele and a wild type Y plus allele. And then we saw vermilion. Really, you could put these in any combination as long as you knew how they're arranged on the parent relative to the offspring. So I've got heterozygous M's and M pluses, V's and V pluses, Y's and Y pluses. A little bit like that was diagrammed on the last illustration. And the assumption is that crossing over kind of happens randomly. So all throughout this chromosome, might be a lot of crossing over events. And they're distributed more or less evenly along the chromosome. So crossing over happens when pieces of DNA that are the same get exchanged with each other. And there's a whole lot of same DNA across a chromosome. So this is going to happen kind of randomly. And as we saw, if crossing over happens between Y and V, then we'll see recombination. In fact, we might see more than one crossover. And in this case, I got 1, 2, 3 crossovers in the space between Y and V. So in the amount of time that I got 3 crossovers there, I got 1 crossover in the space between V and M. So it's just less likely. And maybe you've exaggerated the number of crossover events that would actually happen on this chromosome, but I hope to at least give you the impression that it's more likely to happen between these bigger distances. So between Y and V is a big distance, more likely to have crossing over there than in this little distance between V and M. All right, so like I said, this is a way you can map the locations of genes. So for example, if you didn't know where V was located, you could find out that V was, so basically by the relative distances of two genes relative to a third one, you can decide if it's between them. For example, if you knew about B and C here being 23 units apart, then you could kind of piece that together and say B and A are a shorter distance apart and A and C are also a shorter distance apart. That must put A in between B and C. And then you basically locate a new gene on the chromosome where you didn't know where it was before. So we can use data from controlled crosses to calculate out these genetic distances. And we got a plan to do this in the lab. Kind of work our way through one here from start to finish. So we're doing an example here of a fly with purple eyes and a fly with vestigial wings. And so these are going to be genes that are located on the same chromosome. So first question is, what are their genotypes? And if they are located on the same chromosome, how do we write those genotypes? So purple PR and vestigial is VG. And as we've seen with other fruit fly notations, right? So the purple is homozygous. So if it was unlinked, we'd have something like this: PR, VG plus VG plus, and over here, PR plus VG, VG. But we are linked here, located on the same chromosome. So instead, we want to use the notation that tells us that PR and VG plus are on the same chromosome, PR comma VG plus. So we still have a homozygous individual, but then these are the different homologous chromosomes. So PR, same chromosome as VG plus. I wrote it in this more handwritten version with the flat horizontal line, but that's the same notation as PR comma VG plus, PR comma VG plus. So I think I'd probably have only one option that looks like that. We could also do the other fly, so PR plus VG, PR plus VG is what this looks like here too. All right. So we got their genotypes established. And then they are spelled out here now. What are their F1 individual offspring going to look like? So if we determine their offspring, we should determine their gametes that they can make. So PRVG plus is the same kind of chromosome as PRVG plus. So that's really one kind of gamete this parent can make. Same thing over here, homozygous individual, one kind of gamete. And now the notation becomes extra important here. So PRVG plus, PR plus VG is one way to be heterozygous, and that's the way this offspring would be. And it's different from some of these other options. So we need PRVG plus on one chromosome, PR plus VG on the other chromosome. These other options have a plus and a plus, so that's not even the right combination. This is a homozygous individual again. This one has them on opposite arrangement to what they actually have, and this one's another homozygous individual. So A is the correct answer here, and that's going to get put onto our next slide as we carry this forward. Test cross. I'll just maybe simplify this down and pick the right answer. So a test cross, if we were crossing the F1 individual, would be a individual that's entirely homozygous recessive. So that would be a fly with purple eyes and vestigial wings. If this individual makes gametes, which ones of the gametes would be recombinant? So again, you can kind of maybe start visualizing what the chromosomes look like. The non-recombinant would have the combinations that we see in the parent. So PRVG plus is one of those, PR plus, comma, VG is the other one. And the recombinant gametes would be PR with VG or PR plus with VG plus, right? So if we had chromosomes here crossing over PR, VG plus, PR plus VG, if crossing over happened at some place between them, that would combine PR with VG or PR plus with VG plus. Okay, so let's say we have this as our result. We did this fly, the F1 fly by the homozygous recessive. All these genotypes have one of the chromosomes that's all recessive. And then these are the possibilities. We've already determined that this kind is non-recombinant. This kind is non-recombinant, and this one is recombinant and recombinant. I'll be honest that basically based on something we'll see in a couple slides from now, you can kind of check yourself and say that the non-recombinant, if anything, is disproportionately large. The larger category should be the non-recombinant. I think this is a reliable thing we can expect. So we have in this example, this is just made up numbers by me, but a much smaller number of these kinds of outcomes, and that's because they result from crossing over. All right, so let's get into the calculations for our genetic distance. So genetic distance is calculated as the number of individuals that are recombinant of the offspring over the total number of offspring. So that's 2839 is the sum of all these values. And that turns out to be 0.1074 or 10.7%. So 10% of the offspring are the result of crossing over either to make this PRVG gamete or to make this PR plus VG plus gamete. And it's a relatively small number of the offspring. It's certainly not equal to the number of non-recombinant offspring. And so that's what gives us this 10% crossing over. So you get a chance on one of our assignments to do one of these problems for yourself and see if you can work your way through it. I mentioned earlier that genetic distance is kind of proportional to physical distance, generally speaking. There's not exactly one-to-one. So this is a map of an organism. This is a fungus. And the physical map is the distance among the genes on here in nucleotides. So the units here, MB is mega base pairs. 1 million base pairs is about that distance. And so if you had the DNA sequence of this whole chromosome, which they would have, they could tell you exactly how far it is in nucleotides from one gene to another. And that's something we can do a lot more easier these days, a lot more easily these days. The genetic mapping shows that the order is preserved. So all these same notations for the gene regions are on there, and they're in the same order, but there's not always the same match with some interesting exceptions. So like here, this distance in 16(1b) to the next one, 4-157, it's a very big genetic difference, genetic distance, meaning that crossing over happens quite a bit there. So that's the scale bar. This is about 30% of the time crossing over happens between these locations, but they're not very far apart. So I'll just say that there are mechanisms that can influence whether crossing over happens between two genes, and there's situations that make it more or less likely for crossing over to happen. So there's a reason to use either of these approaches, and they both have their value. All right. Another thing that's one of our last topics here is that there's an upper limit to the amount of crossing over you can observe. So you might, for example, here I've chosen a distance that's about 75 centimorgans or 75% chance of crossing over. Means that crossing over is going to happen in that space about 75% of the time. So that's pretty substantial. And over that distance, the problem is that over big distances, it's more likely that crossing over is going to happen more than once. So if it's 75% likely to having one crossover event, then it's going to be also fairly likely to have more than one crossover event. So I came up with this illustration here. This is for relative distances between two genes, the chance of one crossover event versus no crossovers versus 2, 3, and 4 crossovers. So at a distance of 10% crossover, 10 centimorgans, there's 10% chance of one crossing over, and there's like a 1% chance of no crossing over, or sorry, two crossing overs, and a pretty big chance of no crossovers. So the amount that you would calculate is pretty close to that 10%. But moving up in distance, if you have 20% chance of one crossover, then you get a bigger chance of two crossovers. 30% chance of one crossover gets you a bigger chance of two crossovers, and so on. And eventually, just for convenience, I think it's easy to think about 50% as kind of this cutoff here. And what happens around 50% is that the total of even crossovers is about equal to the total of odd crossovers. So even crossovers, as we saw before, doesn't look like recombination, puts the genes back in the same organization that they were before crossing over. And then odd puts them in a recombinant arrangement. And so in this case, the red plus green is recombinant, and that's about equal to the purple plus orange plus blue. And at 50% crossing over or not crossing over, that actually ends up looking like there wasn't any linkage at all. So maybe the take home from this is that genes can be so far apart that even if crossing over, or even if they're linked, there's so much chance of crossover between them that they're effectively not linked anymore. So you wouldn't really be able to map them very well, but maybe in a composite map, looking at lots of genes on the same chromosome, you'd still know where they were located on there. All right, so lots of topics there. That's our autosomal linkage topic.